Hello, everyone. My name is Shen Li, and I'm the Director of Imaging at Life Canvas Technologies. Thank you all for coming for this webinar. This is our first webinar hosted by Life Canvas, and we're excited to use this opportunity to introduce our new light chain microscope, DALISPIM. It is our great honor to have Dr. Clarence Yep from Peter Soger's group at Harvard Medical School in this webinar. With the generous support from Massachusetts Life Science Center, its device program, Professor Soger's lab at HMS and Life Canvas have established a formal collaboration, and we installed our very first DALISPIM at Soger's lab just a few weeks ago. Beyond this collaboration, Clarence is also a great friend of mine. You can already tell how fun he is from this photo that we took together, also Alex here. The week after the installation, I walked into the lab and saw Clarence prepare a big banner, the balloons, the ribbon, and also a pair of oversized scissors. It really felt like a grand opening for the DALISPIM there. For the webinar today, Clarence will first talk about highly multiplex 3D imaging in human FFPE tumor tissue, and how DALISPIM will fit in their workflow moving forward for this project. Following his talk, I will discuss how the DALISPIM open-top light chain microscope is designed for scalability. Following both presentations, we will hold a Q&A session, and I encourage you to submit any questions you may have during the presentation. Without further ado, Clarence, the floor is yours.
Well, thank you, Shan, for that kind introduction. Actually, those scissors were a pain to bring on my flight through the airport, so I ran with them instead. Anyway, it's a privilege to be invited to speak at the launch event of the DALISPIM, and congratulations to you and your team on this product launch. For those who don't know me, my name is Clarence, and I oversee the advanced imaging projects at the Laboratory of Systems Pharmacology, which is headed by Professor Peter Sorger. Now that just means that I've been using microscopes for the last 2 decades, but I think it's in that capacity that I've been invited to speak about how we're combining high-plex spatial profiling on clinical samples with 3D imaging. In this talk, I will also describe what can be observed, especially in clinical samples. And finally, I'll give some insight into protocol suggestions that we've had to deal with during the development of this protocol. But first, I need to talk about how we are doing spatial profiling. We developed a technique called cyclic immunofluorescence. Normally, only 4 to 5 fluorescent antibodies can be imaged on any given sample before there is serious crosstalk. CySIF gets around that by repeating steps of staining, imaging, and bleaching. Each cycle adds 4 to 5 protein markers and can be done in less than a day. People have found that the protocol has lots of advantages due to its more affordability, open access, and scalability. In fact, if you can scan this QR code, you can obtain the whole protocol here. So previously, people would have been able to image the nuclei with classical DNA dyes. Then they can also stain for tumor cells and their lineages. Then blood vessels. By this point, you'd probably want to figure out where all the immune cells are in relation to the tumor. But normally it would be impossible to further subphenotype each immune cell type without some highly multiplexing technique. It's important to remember that all of this protein information is from the same sample. We can routinely do 100 markers on most samples now by also staining for antibodies specific for extracellular matrix, epigenetic marks, cytokines, and DNA damage. And using CySIF, this can be done on a batch of 50 to 100 traditionally cut 5-micron tissue sections, if imaging by 2D. So how do we go from taking the 2D single Z-plane protocol to 3D? 3D imaging is not a new technique. Confocals and 2-photon imaging has been around for decades, and it's frequently used by the cell biology and neurobiology fields. But something happened in the year 2018. The National Cancer Institute funded a new program called the Human Tumor Atlas Network, a consortia of many institutes across the US to profile cancers of various tissue types. That same year, the NIH also funded HubMap. It too called for the generation of 3D high-resolution atlases. And to date, there are thousands of image data sets of human tissues to study. Admittedly, some of them are 2D though. So So Peter Sorger's lab was one of the first to publish 2 high impact factor publications featuring 3D highly multiplexed data. This well-known Cell paper, with deep collaboration from Professor Ken Lau at Vanderbilt University, analyzed a colorectal cancer specimen that was cut into 25 serial sections and then reassembled. The results showed for one of the first times the shape of a tertiary lymphoid structure across a large scale, and that they are not separate colonies, but connected to each other. The second paper in Cancer Discovery described an early melanoma and invasive melanoma region from the same patient sample using highly multiplexed imaging. 3D high resolution demonstrated that macrophages extended processes towards T cells and to inhibit them via the PD-1 and PD-L1 pathway, and this could explain the prevalence of disease in this patient. These 2 papers do provide 40 to 60 plex, but have limited axial resolution from using thin and discontinuous sections. Now, these are not the only 3D studies in clinical samples by any means. Recently, there have been other landmark efforts by the labs of Professors Dennis Wirtz, Pei-Sun Wu, Ashley Kaiman, Andrew Song, and Faisal Mahmood. The datasets cover huge ranges along the axial axis and cover many more cells. Some are at fine axial resolution too. These are mainly histology or virtual histology papers and make significant headway without relying on large fluorescence antibody panels. But this means there is a method that is missing in between. So therefore, we asked ourselves, what would happen if we combined high-plex imaging with thicker samples beyond the traditional 5-micron thicknesses? So on the left, we cut a traditional 5-micron section of skin tissue and stained it with hooks and did 3D confocal imaging. The nuclei are cut in incomplete slices, as you can see, and the cytoplasms of these cells would be even more incomplete. But then on the right, we took a 35-micron section from the same tissue block in roughly the same area, and by increasing the thickness by 7-fold, we now can start to see complete nuclei and even multiple cell layers thick to do neighborhood analysis along all 3 axes. It's not just nuclear volume measurements that improve with tissue thickness. We asked ourselves if there were improvements in cell classification. Immune cells are challenging because they have many markers which are punctate into diffraction-limited spots and polarized. I'm referring to markers like LAG3, PD-1, granzyme B, and TIM3. If you only cut a portion of an immune cell, how do you know whether you've captured all of its contents if the marker is not spread evenly throughout the cell? Here is a good example illustrating this using real data. You're looking at a dendritic cell flanked by 2 magenta-colored T cells, but from the side. Each of these cells are labeled with typical markers such as CD8, CD11C, PD1, MX1, and LAG3. The last 3 are punctate and polarized. Here, I've simulated what would happen if I cut a traditional 5-micron thick section. If I happen to cut 5 microns here, I'd get remnants of the dendritic cell and a portion of the T cell. I would not have seen that it was activated by PD1. In the second section, I get the most accurate composition of markers, but I'm not seeing the third T cell. Now in the third case is really interesting because it now looks like the dendritic cell is LAG3 positive, which is contradictory to scientific literature. And when we quantify the misclassification of cell types, it can be as high as a third depending on the marker. I want to now demonstrate 3D imaging on thicker samples now in a real case study. The next data will be from a 62-year-old patient who came in with melanoma. Here's a side view of the patient's tumor. It actually isn't the tumor bulge that is interesting. It's those 2 regions on the left and on the right. The left is a melanoma in situ, the very early stages of melanoma. And then just 1.5 centimeters to the right of that is invasive melanoma, where the tumor cells are beginning to transform and invade vertically towards the lymph nodes. We used 3D cyclic immunofluorescence specifically in these 2 regions to see what differences we could identify. We stained and imaged for over 50 proteins. The first thing that is typically done to quantify an image like this is to count the number of different cell types. Skin typically has an epidermal region and a dermal region, separated by the dermal-epidermal junction. the number of melanocytes along the DEJ is unusually very dense here. In contrast, here is a piece of healthy skin from the same patient. Notice how much sparser the melanocytes are here. It's like 1/10 or 1/20 of the cell density as the early melanoma. Our analysis does not need to be limited to just tumor cells. This is high-plex spatial imaging after all. We can also peer into the dermal region as well and look at its rich collagen and vascular network. With this level of resolution, we can also perform vasculature length measurements and observe immune cells extravasing out of blood vessels or within. So earlier in my talk, I demonstrated how 3D imaging can resolve small punctate spots inside cells. But what if we could also gain cell shape information? Looking at cell shape is not that crazy of an idea. When clinicians examine tissues using histological stains, they are primarily looking at shape and texture. Here are 3 areas of a tumor showing muscle, an organized healthier area, and a disorganized tumor region. Clinicians undergo a lot of medical training to visually recognize these as part of diagnostics. So surely there must be something useful that we can learn here to incorporate into our analysis. Let's turn our attention back to our familiar melanoma sample. So we segmented the melanocytes in the early melanoma region, and it's been known from clinical imaging that melanocytes in this area have long dendrites. But as they transform and leave the dermal epidermal junction to a more hostile environment, they actually round up. They also round up and lose their dendrites in the invasive melanoma region too. It's not just tumor cells that express different shape. Check out the change of shape of cytotoxic T cells in different stages of melanoma. In early melanoma, cells are round, but towards the invasive melanoma, they start to develop these long tails. And then by metastatic melanoma, the immune cells have gained these very long microvilli. And these features can give clues as to the state of disease and their immune state. But unfortunately, this intricate feature has not been widely used in conjunction with protein level yet. So to demonstrate that there is some value of analyzing 3D cell shape, I trained a model to extract features from thousands of cells and represent the features of all the T cells in T-SNE space. Each dot that you see in the middle here represents a single cell. Now the model was blind to which melanoma region these T cells were from. Only I knew where they were from. Yet when I overlay their ground truth as different colors, their positions appear to cluster, suggesting that the model has learned some relationship between cell shape and disease. It would be interesting to inspect the cells that overlap near the boundaries of these clusters and see why they are some of the first of their population to change shape. Who are their neighbors? Are they attacking tumor cells? Are they becoming more activated by antigen-presenting cells? These are some of the questions that interest us and are relevant to many diseases. When I first started my talk, I showed how small punctate spots could be misassigned to cells, leading to wrong cell type classification. But I want to discuss this more because we now have higher resolution imaging capabilities, and so we should really capitalize on this. We see PD-1 and PD-L1 engaged towards tumor cells. We can see granzyme B directed from immune cells too, and this can assist in our detection of active cell engagement. Now, in the invasive melanoma region, we see thousands of PD-1 and PD-L1 puncta between pairs of T cells and dendritic cells. Can you guess how many interactions there were in the melanoma in situ region? Just one, and it's between this T cell and tumor cell. So the third thing you can do with the added resolution from 3D-Decisive is to look at which cells are communicating with each other. Within a community of cells, we can have a variety of cell types. Some of them may be interacting with each other, while others may happen to be just passing by each other. So how might we go about assessing and verifying membrane-to-membrane contact then? We can draw line profiles across the membrane and look for the overlap of the peaks. We're calling a junction between two cells as tight when the separation of the peaks is within tens of nanometers after interpolation. A loose junction, where cells may just be passing by each other, has a separation on the order of microns. So let's try to apply the three features we just heard about together towards a problem. Here is a T cell in close proximity to a tumor cell. Because we have high resolution 3D imaging, we can resolve all the punctus stains, in this case granzyme B, in this T cell. This T cell also happens to be directing a granzyme B particle towards the tumor cell. Is the membrane of the T cell engaged with the tumor cell? Yes. The separation is around 50 nanometers, which lines up nicely with literature of immune synapses imaged by electron microscopy. And finally, we have the shape of the T and tumor cell. This cell appears to be quite spiky, and it has microvilli sticking out. Therefore, it's conceivable that we're looking at a metastatic melanoma. Okay, that was just between two cherry-picked cells. What if we looked at a community of cells? How about this one? Again, in metastatic melanoma. We see here that if we pull back a few layers of cells, it reveals a few effector T cells interacting with each other, again with those long projections. We see that one of the T cells is engaging with the tumor cell because it is directing its granzyme B payload in green towards it. Because we have 3D imaging, we can see that this T cell is simultaneously in contact with a CD11C positive antigen presenting cell, but counterintuitively is being negatively regulated via PD-L1, shown with green dots. Since this pattern is not well understood or characterized yet, it would be fascinating to understand what is happening here and how that impacts tumor resolution.
But that's only a small community of cells. Have you done it on the whole tissue yet?
We have, and we deployed this to another cell type that is a hot topic right now. Exhausted progenitor cells, or TPEX cells, are trendy because they give rise to new T cells that can combat disease. We needed some additional support, so we got help from Professor Gaudens Danuser, formerly at UT Southwestern, and his postdoc Felix Zo, who developed a more accurate 3D segmentation tool called USegment 3D, now published in Nature Methods as of 2025. Using this tool, we identified all T cells and TPEX cells and found that very few of them, less than 1%, were attacking tumor cells. The majority of them were not in contact with tumor cells at all, since their role is thought to be more renewal of T cells. Just to demonstrate that this technique also works in other diseases and tissue types, we've applied 3D CISIF imaging to ovarian cancer, glioblastoma, and colorectal cancer as well. So hopefully I've convinced you to give 3D CISIF a try on a microscope that you might already have access to via Core or your own lab. The instruments used in this work was a Zeiss and Leica laser scanning confocal that was owned by a microscopy core. I want to go over a few technical considerations to get the best data possible. The most important one is not what you think. It's the stage insert. Most of your samples are going to be on slides and imaged between cycles, so it's important that they are consistently placed each time they return for the next imaging cycle. Stage inserts with sliders and no clamps are to be avoided as they still permit some rotation and translation. The most frequent question I get is, "Hey man, the imaging is too slow. Can you make it any faster?" Well, sure, laser scanning confocal microscopy is painfully slow, but you get excellent contrast in thicker samples. We also attempted spinning this confocal and found that images were indeed comparable to laser scanning confocal, albeit with some flaring of the point spread function. More importantly, there was a threefold speed improvement, which meant that we could cover threefold more specimens or volume. When it comes to selecting primary conjugated antibodies for cyclic immunofluorescence, some thought should be put into selecting optimal fluorophores. Tissue autofluorescence exists mainly in the blue and green wavelengths. Therefore, it is better to avoid putting weak expression targets here or until after several cycles when autofluorescence would have been reduced by bleaching. Cameras have poor sensitivity in the near infrared range, and so putting weak targets here should be avoided. For fragile tissues, you may see your sample disintegrating or moving. This is detrimental to downstream analysis, and so in this example, this piece of tissue has developed a large hole, and if the damage becomes too severe, then it might be necessary to identify more stable tissues, and sample mounting continues to be a subject of continued development. So what's next on the horizon for 3D imaging, and how often do we need it? Right now, no one is suggesting that 3D will fully replace 2D imaging. 3D is still quite expensive and requires more patience to set up and maintain, although I know that LifeCanvas and other companies are working to change that. Image data files are orders of magnitude larger, and analysis is slower. But what I have hoped to show you is that 3D can offer deeper as well as complementary insight to existing profiling methods. We showed that high-resolution confocal is needed when the subject matter is larger than the tissue thickness itself. In our case, this was 35 microns. However, there are a lot of biological phenomena that exist beyond 35 microns. Inflammatory domains labeled with MX1 appear to be isolated here, but could they all be connected if viewed in a thicker sample? Blood vessels and nerves are very long, on the order of centimeters or meters. Lymphoid aggregates are hundreds of microns wide and tall. Colonic crypts are also on the millimeter scale, and so should the thickness of the specimen. Therefore, to capture the heterogeneity in such a thick specimen, we must turn to another form of 3D imaging, which is light sheet. We recently used the DALISPIM from Life Canvas Technologies to look at thick colon specimens. The ones you see here are healthy specimens, but diseased ones can be imaged too. As expected, we see Claudin 7 stains for the colonic crypts. The crypts themselves contain intestinal stem cells at their bases and stain positive for Ki67. Tub 3 stains for nerves, and they look like what they're supposed to look like, and between the crypts. It's been known that the colon contains lymphoid aggregates, which are immune cell rich, which we see very nicely with high contrast here and are easily identifiable based on DAPI alone. There is already much work being done on applying cyclic immunofluorescence on understanding the structure and the composition of these immune aggregates and how it relates to forming a protective defense system against diseases in lung and colon. We had the fortune of testing various optical designs of light sheet. We have found that certain specifications are more beneficial to our samples. Most of the specimens we get are cut in such a way that they are flat and wide, but thin, around 2 millimeters thick. The way in which we mount this seems to benefit by having an inverted design where the sample is scanned horizontally from left to right or right to left. Since the samples are typically a few centimeters in width and length, a large travel distance is not really needed. Instead, it is more important for us that the stage travels consistently and is accurate in movement. Sensitivity to signal is also important for this kind of work because we use primary conjugated antibodies, which are generally dimmer. Therefore, having the most sensitive SCMOS camera paired with oil immersion objective lenses with long working distances is needed. Other features that are crucial are having multiple laser lines and filters and the ability to support multiple refractive indices. And finally, we need non-proprietary file formats that can plug and play into existing or new image analysis pipelines. How might the future look like for 3D imaging, specifically clinical samples? Well, one such tool that is already very popular is the ability to generate pseudo or virtual H&E images that are familiar to clinicians. This is generated from a fluorescent nuclear stain and tissue autofluorescence. This is becoming a valuable procedure to do because it gets clinicians engaged with our research to help us identify interesting biological questions in healthcare. Why am I bringing this up? Because 3D optical and serial reconstruction are the only way to get this kind of clinical purple and pink image from a 1 millimeter thick dataset. And to my knowledge, you cannot get this using traditional bright field unless with some sophisticated instrumentation. And so let's apply this to our favorite melanoma sample. It should be pointed out that in the virtual H&E image, all cells are now more visible with some contrast, even if we did not have a specific stain for those cells. And the way we detect cells in fluorescence, on the other hand, is by being deliberate with our antibody choices. Otherwise, this cell type would be invisible. So we've covered high-resolution confocal microscopy, virtual H&E imaging, and light sheet microscopy. Can these offer some complementary advantages with each other? And therefore, an interesting concept that has been floating around is to use light sheet imaging to get either a virtual H&E image with a medium number of targets, maybe 10 to 15 channels, and this allows you to identify coarsely where something biologically interesting is happening. This could be immune invasion into the tumor, or where the immune system is interacting with the tumor, but nothing's really happening yet. Once that area is identified, you can precisely section that region out and perform downstream high-resolution 2D or 3D imaging. This could be a more efficient way of using both resolutions, especially in an age where it costs money to store and analyze large datasets. If you'd like to have a look at the data, we have developed a tool called Minerva Stories, which gives you a Google Maps feel to the size of data. This works completely in your browser without downloading any files or signing up for subscriptions and watching ads. You can pan and turn channels on and off, but currently this is limited to maximum intensity projections of the 3D data at this time, but we hope that future versions will have 3D capabilities. Well, before I hand it back to Shan, I have many people to thank, including you, for watching. So the Laboratory of Systems Pharmacology is headed by Professor Peter Sorger and Professor Sandro Santagata. They both provide excellent leadership direction and open new and exciting opportunities. Within the LSP, we have many senior scientists, postdocs, and students. In particular, Alex Wong is a postdoctoral fellow who is developing the protocol for CISIF with light sheet on 1 mm thick specimens. He is joined by Daniel Liu, who is a PhD student who is adopting both light sheet and confocal to address outstanding questions regarding T-cell biology in cancer. So Woo Choi is our newest member to the 3D team and brings a lot of light sheet and sample preparation experience from MIT. And he is jointly supervised by Life Canvas Technologies. A project of this magnitude requires organization of both project scope and data, and so we have an incredible team made up of Elise, Allison, and Sarah. These files are enormous and will only get bigger, yet our mandate is to provide them to anyone who wants them for free, anytime and anywhere. And our software and data team is headed by Jeremy in this respect. I mentioned several times that working with clinical samples requires deliberate input from clinicians, and we are lucky to have experts in the skin and immunology field to help with our data interpretation. 3D imaging is more complicated, and it requires a dedicated core facility team to guide instrument usage and design validation experiments in a scientifically rigorous manner. The Micron facility is headed by Paula at Harvard Medical School and has an amazing team and impressive selection of microscopes. Scientific funding is maybe the most challenging thing the school has ever faced since the 1600s, and so I want to give a special shout out to Audrey Bryan and the team at MLSC for administering such a generous state funding portfolio. We look forward to working with her through the Bits to Bytes program. And last but not least, I want to thank the folks at Life Canvas for organizing this talk, being exceptionally collaborative, providing technical advice within minutes of needing help, and being great colleagues. It is a pleasure to push the boundaries of science with you. In closing, I also want to highlight other collaborators from around the country who have gone on this journey with us. At UT Southwestern, we also work with Professor Kevin Dean and another postdoc, Zigo Sheng. They've developed tools to automatically quantify specific cell interactions in a statistically powered way. Other collaborations we have are with Professor Kari Borner and Niels Gellenborg from HubMap to map out distances in tissue as well as visualize the datasets more easily using augmented reality, respectively. They all have papers highlighting the development of much needed 3D tools that are compatible with the 3D data I described, and more.
Thank you, Clarence, for the excellent talk and also exciting work ahead. So for the second part of the webinar today, I'm going to introduce the DALISPIM, a new open-top light sheet microscope that is designed for scalability and also ready for automation. So a little bit introduction of what we do at Life Canvas. Life Canvas is the pioneer for the 3D spatial biology. We're the only company providing end-to-end solutions for clear tissue imaging from the sample preparation, light sheet volumetric imaging, and also data storage and analysis. And we have the experts in every part of the pipeline. When we're working with Peter Sorger's group with Clarence, we're not just providing the DALISPIM light sheet microscope, we also provide the support and guidance on the sample preparation for the thick tissues, and also the downstream data storage, and also the management. So for our integrated end-to-end pipeline for 3D spatial biology, we have our own technologies, including the reagents and also devices. So for the sample preparations, we have our own technologies for the preservation of the tissues, the delimitation, immunolabeling, index matching for the thick tissues. And for the light sheet imaging, we have our own light sheet microscopes with 3 different models to enable the high throughput, high resolution 3D volumetric imaging. And finally, once you get the large amount of the data, we have the downstream data management, storage, and analysis. And today I'm going to focus more on the light sheet volumetric imaging part. Over the past decade, there are several different configurations of the light sheet microscope, including 2 of our own, and they're optimized for different tissue types or different tissue geometries. So on the left, we have the SmartSpim that is optimized for large intact tissues, and the largest that we can image is a marmoset hemisphere intact. And on the right, we have the Megaspin that is more optimized for a small sample array or a large coronal slabs of the human brain, or like NHP. For those different configurations, they're limited to the scalability, and mostly because of the limitation of the sample mounting. Because for those of the microscope, most of the time you need a very specialized or a custom sample carriers, and it is very hard to integrate in other existing workflows. Other than that, the sample mounting might include like gel embedding and glue, and the protocol for those can be a little bit difficult to handle for larger numbers of samples, like tens or even hundreds of them. So there's a need in the market to have a commercialized light sheet microscope that is designed for scalability. So to scale up the real process, first of all, you need a very easy sample mounting so that you can mount even hundreds or thousands of samples eventually. And second of all, you want a very standard sample carriers so you don't need to pop up the sample from the light sheet and put on another sample carriers for other optical microscope. So if you Google what's the standard sample carriers that is designed for scaled up for the imaging, then most of the time you will definitely get a multi-well plates. So the DALISPIM that we're designing, it is compatible with cover slip bottom multi-well plates to enable scalable workflows. So before I talk about the core technology of DALISPIM, I want to quickly show you the final results. On the left, there are 12 organoids with 800 micron diameters. On the right, there are 12 1 millimeter thick mouse brain slices. So the beauty of those two data sets is that each set of them were clear and labeled and imaged on DALISPIM in a 12-well plate. This capability makes the large-scale automation significantly more achievable. And for the organoids, the imaging time is about 53 seconds per channel per sample. And a few weeks ago, we also tried imaging organoids in 96 well plate, and it worked really well. And now the question is, what's the technology behind it? So I want to be a little bit derailed here from like a normal vendor talk, because I really want to tell you the real story that happened to this innovation. So as I mentioned before, we have the Megaspin with the optics above the samples with this 45-degree angles designed for a small sample array and also a large sample slab. To prevent any aberrations from the air to the immersion, we have to cover the entire light path from the terminal optics to the sample and back to the detection with the immersion oil. But this design is actually limiting the scalabilities because, first of all, you need a very customized sample holder, and also it's hard to swap the sample easily. So I remember it was in 2024 in October, because my wife and I were on a vacation at Disney World, and we're waiting in the long line. And then I saw a Slack messages from my colleagues, Brian and Nick. And it's a 3 people Slack chat. And then they're talking about, oh, there might be a new way, a different way to tackle these problems to make it better for scalabilities. So the trick is that we bag the sample with the immersion in there, and also the bag has the same refractive index 1.52, same as our samples. And also at the air gap here, instead of having the whole immersion pool, we can bag the immersion as well. And also with the cover slip to prevent any air gap between the optics to the sample back to the detection path. And when I first saw this idea, it was very exciting, and it could maybe resolve the issues that we have had and also increase the capability of our microscope. And also during my vacation, Brian and Nick put together this idea into the reality. And at the time, we called it a sack spin. So they basically put this big bag of the immersion oil and with a clip around it to make sure it's not moving with the samples. And you would think that with such janky setup, the image quality at the end would be bad, right? But the result was really good. As you can see on the left, it's the XZ cross-section. On the right, it's the XY. The imaging quality was quite sharp. There is very minimum elongation along the detection optical axis. And that's how we want to move it forward from. So even though this the setup is not optimized, there are several advantages already. Because for this the sack spin or eventually the DALI spin, the light sheet technology eliminates the need for a large immersion chambers, a large immersion pool. And also secondly, the sample is back and also separated from the immersion in a light path. And also it will be a lot easier to streamline the process for the sample exchange. However, there are several key improvements that we have to do because first of all, it's not commercialized. We have to make it ready for immediately lab use. Otherwise, it's very hard to be reproducible. And second of all, instead of having the optics on the top of the samples, we want it to be an open top, meaning that we flip the optics upside down so that we have the full axis from the top to put the sample on, which will enable a lot of the possibilities for the sample exchange eventually. So after 6 months of the design of this bag idea and also a year to fully commercialize the product, we eventually officially launched the product at the end of last year. And here comes the DALISPIM. Our key technology is patent pending DALI technology, stands for Depth Adaptive Light Interface. The key idea is that there's a DALI device that is put between the illumination and detection path and to the samples to prevent this angle sample carrier interface to the air, allowing for this open top configuration. So within this DALI device, it is filled with index matching immersion, and also the deformable interfaces maintains the continuous contact to the sample carrier without any air gap. So when a sample is moving up and down, there's no air gap at all. So everything in the light path, the immersion, the cover slip bottom, the sample back to the detection path, everything has the same refractive index at 1.52. And also with the DALISPIM, we're able to image the samples up to the thickness of 7 to 7.5 millimeters, and no large bath is required. So with the DALISPIM technologies, there are a few key features. First of all, it has a broad platform capabilities, meaning that we can bridge the gap between the light sheet and other imaging modalities. So most of the time, if you use a light sheet microscope, it has its own sample carriers, and you have to take the sample out and then put it on another substrate for imaging on another optical microscope. But on the DALISPIM, imagine that you can image the whole volumetric data with a lower resolution, and then immediately you can move this cover slip bottom well plate to another inverted confocal microscope for higher resolution imaging up to 60x or even 100x, which is the resolution that is not feasible on any light sheet microscope so far. Second of all, it is designed for scalability. It has its open top configuration, and also is compatible with well plate. So in the future, we can implement a robotic arms for the sample exchange with the automation. And lastly, it's flexible across different applications. It's very easy to use, including the sample mounting. Some of the samples, you can just drop it in the well plate and then cover it with the immersion oil, and that's it, ready for imaging. And also it's accessible from the top to take the sample in and out. So on Clarence's case at Peter Soger's group, it's much easier to implement the highly-plex imaging. And also it supports a wide range of modular attachments that you can think of for any inverted microscope. For example, you can just put a heat block on the top of the well plate for environmental control. So here I want to talk more about the features that are shared across of our different light sheet microscopes. One of the features is the axial sweeping. So the axial sweeping is the technology to enable the homogeneous and best axial resolution across the entire field of view and up to the entire volume. The key idea is that we have this sweeping light sheet across the field of view. At the same time, we have the rolling shutter on the camera that is synchronized with the sweeping light sheets. So we only record the signals from the thinnest part of the light sheet to get the best axial resolutions. So this process is continuous for each frame, and that's how we're able to rapidly acquire the volumetric data with near isotropic resolution. So with the 800 micron organoids that we imaged before with the 9x objective in the previous video, we're able to achieve 53 seconds per channel per samples. But we also just figure out that we can basically double the imaging speed. So it's about actually 27 seconds per channel right now. So to show you a little bit of the axial sweeping, on the left, this is the XZ cross-section when we turn off the axial sweeping. As you can see in the entire field of view, only at the center of the field of view has the best axial resolution, while on both ends, it's quite blurry, poor contrast, because the light sheet and the center of the field of view is the thinnest, but it's much more thicker on the side of the field of view. But then once we turn on the axial sweeping, the sweeping light sheet is synchronized with the rolling shutter on the camera, and then we capture the thinnest part of the light sheet to enable the homogeneous light sheet thickness and the thinnest. That's how we're able to achieve such a best axial resolution with the rapid acquisition. And of course, there are some trade-offs of the axial sweeping. Just like any other optical microscopes, you always have to combat with imaging speed, signal-to-noise ratio, and also the axial resolutions. Obviously, you cannot have it all to be the best, but you can choose what matters the most to you. For example, if you have some labels that are dimmer, so you might want to increase the signal-to-noise ratio. So you can either choose to slower down the scanning so the imaging speed will be lower, or you can open this virtual slit, getting more signals from the light sheet, but then in return, you're getting the signals from a thicker part of the light sheet as well, so the axial resolution will be slightly poorer. So this part we can work with you to see what matters and also what makes more sense for your research. And then I want to talk about the specifications. So obviously we can image the well plates, the whole well plates on the DALISPIM. As for the laser line choice, on the base systems, we have our standard laser lines 488, 561, and 640. And optional laser lines including 405, 445, and then up to 785 nanometers. And we can put up to 7 laser lines in the laser combiner. For the 405 laser, however, it is only recommended for the sample thickness up to 1 millimeters because it has a very strong scattering for such a short wavelength. So after 1 millimeters, the signals might be unusable. As for the 785, we probably have tried most of the dyes out there, if not all of the dyes. We only found one probably usable dye in 785, but it is still like very bleachy to be usable. But at the bottom here, we have a data set of the mouse brain slices, including 5 channels from 405 to 690. And we actually have another data set that includes 6 channels, which is what we can do right now in the same sample. As for the detection objectives, we have 2 of the lowest magnifications, 1.8x and 3.6x, and they're able to image the sample size up to 7 millimeters and also 10 millimeters. So for the 10 millimeters, you can image up to like a whole mouse brain with 1.8x. The 3.6x, you can image maybe like a mouse brain hemispheres. As for the higher magnification, like 9x and 15x, the sample thickness could be a little bit thinner, up to 5 or 3 millimeters. As for the software control, because all of our software are designed for light sheet microscopes, it's especially user-friendly. And also for the procedures, we have the guided step 1 to 4 steps, and the users just need to follow step 1, 2, 3, 4, and then that's it, ready for imaging. And also after acquisition, there's a downstream streamlined post-processing workflows. So the final data set will be a stitched data set in TIFFs, or you can also convert it into ZAR for your further analysis. And then also we have the solutions for the integrated data management that is compatible with our entire pipeline. Other than the organoids or mouse brain slices that I showed before, we can actually do more on DALISPIM. We imaged the mouse lung, kidney slice, hemisphere, eyeball, and also we imaged tick just for fun. Or for revenge, my colleague Zach was bit by a tick, and then he took it out, clear it, and image it on the DALISPIM. Other than the standard cover slip bottom well plates, the DALISPIM is also compatible with most of the glass bottom, including petri dishes, slide glasses, organ on the chip, and also we can make a custom sample holders for any of the tissue geometries. We can image up to NHP slabs or human coronal slabs as well. Finally, I want to reiterate the key features of the DALISPIM open-top light sheet microscope. First of all, it has the broad platform capabilities. It bridges the gap between the light sheet and other imaging modalities. And second of all, it is scalable by design with its open-top configuration and also compatibility with the well plates. It is ready for automation. And third, it's flexible across different applications. It's very easy to mount, and it supports a wide range of the modular attachments with most of the inverted microscope. And lastly, it has the high throughput volumetric imaging with the near isotropic resolution, also compatible with the multi-well plates up to 96 well. And finally, we open to any kind of collaborations. And also if you want to learn more about the DALISPIM technologies, feel free to contact me at the bottom email here. And I think this is the time for our Q&A session. Thank you.
All right. So I would still encourage people to put their questions in the chat. You can click on more, and there's a Q&A tab. So maybe people are having trouble finding it in there. When I first used the webinar, I also had trouble doing that. So I saw Michael Weber sending the message to us asking about which 785 dies that we have used. So the one that we used that was kind of working in 785 channel was it was called MB800Z. And also we contacted the author of the paper, and he also mentioned that that might be the best die for that channel for now. But hopefully in the future there will be a much better chemistry of the die in the future. And I got another question from Laurel Schruck. Does DALISPIM require easy index, reflective index 1.52 for immersion, or can you change the immersion media to be compatible with other clearing methods? So that's a great question. So for now, we focus a lot more on the compatibility of the DALISPIM to our immersion, which is easy index, 1.52 reflective index. And we're working on make it work for the other immersion as well. We have tried the 1.56, which is for like iDisco samples, and that worked fine. But we're trying to make it work better by modifying maybe the cover slip bottom. Instead of using the normal glass 1.52, we might need to glue a different glass that is matching to the reflective index of the iDisco samples. And also we're actively working on the improvement to make it compatible with water as well, because I know some people want to image maybe like expanded tissues or live organoids as well, which by the way, for the live imaging, the limitation of the sample, the imageable sample depth is going to be limited by the scattering. So maybe you can image up to 100 micron-ish. Depends on the density of samples as well for the live samples. But after a certain thickness, you still need to clear it for better penetration, also better labeling afterwards. And from James, very exciting. Does each objective use the same RI medium? How difficult it is to swap out the objectives? Yeah, so I already briefly mentioned the RI exchange. As for how difficult it is to swap out the objectives, we have designed the objective to be mounted on the magnetic mounts. So on the software, you just click on dip. So the objective will be extracted from the microscope, and then you just basically just take out the objective immediately, like rotate it a little bit and take out objective, and you can take another new objective in there. And it's really smooth. It maybe take you like 10 seconds. Of course, you have to clean the objective OEO from that. And I actually have a question for Clarence. I'm just wondering how, as you know, for we have a selection of different objective with different magnifications. And people know that for the light imaging, you're choosing between higher resolution, but also like a large volume of the data, versus like the lower resolution part. Like, how did you choose between the objectives on the DALISPIM and also compatible with the workflow with the confocal?
Oh, dang, that was a question that I was going to actually ask you. I think we obviously had a lot of discussions prior to the purchasing of the DALISPIM, and we were very happy with the broad range of magnifications. I think what we wanted was something that was not too much overlapping with the existing microscopes that we had in the lab. We wanted something that was very orthogonal, that would not compete. And so, as I described earlier in the talk, we have a confocal with oil immersion objective lenses at 40x and 63x magnification. But we didn't really have anything that was a little bit more lower magnification that would help complement that. And so I think when we reached out to you, you recommended the 9x, and I think that was a really good selection. I know that you also have a 15x in your brochure. That would have also been really suitable as well. But we didn't want something too high resolution just yet, because we wanted to see the whole the tissue, and maybe once we get as the field moves on, it would be nice to then switch to higher magnification to see what we can get. And I think so. Therefore, there's lots of room to have higher magnifications. Actually, I had a really naive question about working distances, and so our samples are getting thicker and thicker, a millimeter thick. So, really naive question: is it really common for all of these light sheet objective lenses to have a millimeter or greater? What's the largest working distance that you can have on some of these lenses?
Yeah, for the DALISPIM, because we have this 45-degree angles relative to the samples, so we actually compromise a little bit, sacrifice a little bit of working distance for the ease of use of these configurations. So for 9x, the one that you're usually use, we can image up to 5 millimeters. For the 15x, we can image up to about 3 millimeters. We're working on the different versions to basically rotate the optics. So eventually we might be able to make it compatible with 22x, but that might be like later this year down the road. We actually got another question from Michael Anderson. This is for Clarence. When you do multiplex imaging, are you using clear samples? If so, can you all restain and bleaching in RA matched media?
It's a really good question. Sorry about the ambulance outside. Somebody's hurt, but yes, it's a really good question. Yeah, no, we're not able to stain while it's in refractive index matching. We have to go back and forth between aqueous buffer and then stain, and then re-clear again. So it adds about an extra day to the whole process. And if I know, Michael, you're just down the road from us. If you're interested, I can put you in touch with Alex Wong, who's the postdoc who's specializing in this protocol.
Okay, and we got another question from Michael Weber. This is also for Clarence. How will you go about labeling thicker samples for the DALI with antibodies considering their limited penetration performance, especially considering you want to multiplex?
It's a great question from Michael, and it's good to hear from you again, Michael. Yeah, we work with LifeCanvas to develop our protocol, and they have their smart batch stochastic electrotransport device. We purchased one in the lab after extensive demoing, and I mean, Shen can talk more about how it works, but for a lot of antibodies that have very poor performance in terms of penetration, they need this extra modification. It takes about 25 hours. We have also come across a lot of antibodies that are able to passively diffuse overnight. It takes about 18 hours at room temperature, but those samples have to be a little bit thinner, more like 300 to 400 microns. So some antibodies can and some antibodies cannot. And I think one of the challenges to doing this project is the amount of resources required to validate these antibodies. It's an ongoing question. Yeah. Well, two questions from two Michaels that I know, so that's great.
Okay, another question for me from Wu Yu. What was the best resolution you could get with the organoids? So the highest magnifications that we have on the DALISPIM is 15x, and the voxel size for that would be 0.4 micron-ish. But the axial resolution along the optical axis would be around 2 micron. But we might be able to toggle the axial resolution, like I mentioned before, if you're okay with compromising a little bit of the speed or the signal to noise ratio. So this is something that we can work with. I think that's all for the Q&A today. Thank you all for coming, and especially thank you, Clarence, for joining this webinar.
Thank you for the invitation, Jen, and to your team, and congratulations on your product launch.
Yeah, thank you. All right, I think that's a wrap for the webinar today. And oh yeah, by the way, for all of the participants, you will get a download afterwards. Yeah, and we'll for sure post more on our social media. Thank you.